And welcome back, everybody. So this is session 2 of SNUPA 2021. And it's my great pleasure to introduce to you our first speaker of the session, which is now a little bit more focused on computational neuroscience and neuroscience questions. And our speaker is, who is, I'll just mute him for a moment. So, I guess some echo somehow that I'm kind of fighting at the moment, but okay. Sorry. Okay, good. So I think now that should help. So, great. So Henning is basically somebody who's been working for a long time on spiking neural networks and questions pertaining to plasticity and learning. And he did 2 very successful postdocs with Wolfram Gerstner and Richard Kempter. He had a stay as a professor at the University of Cambridge, and now he's at the Technical University in Berlin, where his lab very much still focuses on learning in spiking neural networks or neural networks in general, and also most recently get increasingly interested in inhibitory interneurons and the role of inhibition in these networks. And I'm very much looking forward to your talk, Henning. Thank you very much for coming. And without any further ado, please take it away.
Good. Thank you very much, Friedemann, for the nice introduction. Thanks a lot for inviting me and for setting up this beautiful workshop. Maybe as a starting point, this is truly a lot more biology than in the last session. So I'll torture you with loads of weird facts about the cortex. So we've come to spike for the optimization of spiking networks through sort of a completely different path, namely trying to understand how cortical circuits work. But in a way, the motivation that took us there is very much linked to the one that Tara mentioned at the beginning. It's the one of parameter tweaking, right? Because that's something that's a curse, and many of you will be familiar with that. And if you actually just build models of cortical circuits, you very quickly run into that type of problem as well. Good. So first, maybe I should say that, of course, this is not really my work in the end. This is very much the work of Joram Kaiser, a PhD student in my lab, who's just done amazing work. And Richard Naud, who's now, of course, running his own lab and doing amazing stuff over there. But I had the luxury of actually having him as a postdoc for a while, working with him as a postdoc in my lab a couple of years back. So as I said, we're interested in cortical circuits. And so I thought I'd just shock you with this type of image for the beginning. So what you see here is in the center, the good old friend that we all know is the pyramidal cell, the sort of the almost emblematic cell of the cortex. And those guys are in cortex, about 80% of the cells. They're excitatory, and they're comparatively homogeneous from cell to cell, although that's kind of starting to crumble as well. And the other 20% are all these colorful guys that are hanging around here. And the reason they're so colorful is that they're basically evolution's vengeance for the simplicity of the pyramidal cell. They are super diverse. Cells look very different. They do very different things. They're electrophysiologically different. They're different in their protein expression. They're different in where they sit in the cortex. And it's just a bit of a mess, right? So it's really hard to understand what they do. Now, we get more and more data on that. But, reasoning through complicated circuits is something that, humans are notoriously bad at, I think. And so it kind of calls for a model. So that's what we embarked on doing. So, because I presume that many of you are more familiar with sort of the simpler or the more engineering side of it all, let me give you a very quick rundown of what these different interneurons are, in which ways they're different. And one thing that's very striking is that they target very different places within the circuit. Okay, so basically, if you look at one of such a pyramidal cell, then the sort of textbook idea is that they receive bottom-up input or sort of sensory drive around their soma, and then they have these beautiful intricate dendrites to the top, and up there in layer 1, they receive top-down information from other cortical areas, things that are very much pertinent to learning, etc. And so what these interneurons now do is that some of them very much target the soma. Those are cells that often express parvalbumin, and they're well suited to suppress spikes. A different type of cells targets the dendrites. Those are these light blue guys up here, and they are that predestines them to suppress sort of active synaptic integration or dendritic integration up there in the dendrite, and thereby alter the integration of top-down signals. Something that I would like to highlight, because I'll come back to that, is that these cells not only differ in where they project, but also what kind of input they receive. So the cells that go for the soma often receive inputs or synapses that are short-term depressing, meaning if you have a spike comes right afterwards on these inputs, and the second spike causes less of a result in the inhibitory cell. And that's very different for the ones that target the dendrites. So you have these somatostatin expressing positive cells expressing cells that target the distal dendrites, and they often have facilitating inputs. So if you have two spikes in quick succession, the second spike is often a lot more effective at driving these cells. Actually, quite strikingly so. Sometimes the first spike is just silent. So that's something I'll come back to later on. Then there are other types of cells, like these weird NDNF cells, neurogliaform cells sitting in layer 1, and they do something interesting. They just release bulk GABA into the extracellular space. And it's known that at least partially they also target sort of presynaptic terminals of other cells, or sort of presynaptic terminals that target, for example, the pyramidal cells, and thereby suppress synaptic release. And finally, there are yet other interneurons that basically target not the pyramidal cells, but they target other interneurons. So there's sort of a meta level where you don't have inhibition, but disinhibition or disinhibition, and you can sort of take that arbitrarily high. Okay, and I think the thing that you'll all see is that this is a pretty complicated system, and complicated systems are hard to understand. So maybe one should try to set up some kind of computational model for that. And that's what we wanted to do. We started with that probably 5 or 6 years ago, and it was initially a nightmare, right? Let's put it that way. And so I'll focus a little bit on a simpler circuit for the time being, but even if you take a comparatively simple circuit, you very quickly run into a problem that these circuits have many, many parameters, right? And you can imagine that if you have n different cell types and you connect them all in different ways, then each of these connections basically needs some kind of strength. So, and the number of these synaptic pathways basically increases like n squared. So even if you just have, 3 cell types like you have them here, or maybe you have to worry about the dendrite as well, then it's 4, you end up with something like 16 parameters only just for the connectivity. So that's something that just very quickly explodes, and that makes bottom-up modeling, basically just sort of, taking the data and then trying to build up a phenomenological model of that extremely hard and extremely painful. And the take home that we had is that you can make these things do whatever you want, right? You just choose the parameters and then they just do stuff. And you can make them do the opposite as well if you choose the parameters the other way. And so that's, of course, sort of an obstacle if you try to understand what they do. And so the alternative that we started to take then at some point is to just, optimize the circuits. Take those parameters that we don't know and then just optimize them by gradient descent, right? And then the resulting circuit that you get out is then something that you can compare to experiments and see if you can explain basically the data in terms of some kind of objective function or some kind of computation. And so this is really as I said in the beginning, this is very much sort of the approach that Tara has argued for. Let's get rid of every single parameter tweaking that we need to do. Let's do all of this by plasticity or optimization. The problem, of course, that we face then is that we don't have the luxurious situation that you have in engineering, that you know what you would like to do, right? We're in biology, so we have these cortical circuits, and we have no idea what they really do. That's actually what we want to find out, right? So we have a bit of a chicken-egg issue here, that we don't know really what the circuits look like, but we also don't know what they actually do. And so we have to make some kind of arbitrary choice here. And so the starting point that we chose is one that I personally think is a bit of a straw man objective. And I don't quite know why this should be an objective at all, but it has turned out to be surprisingly successful. And that's really just optimizing these circuits for a balance of excitation and inhibition. I'm very happy to chat afterwards why this could be useful, but let's just use it and go for it, okay? So excitation inhibition balance is something that's found very broadly in cortex and also in many other areas. So it's known for quite a long time that if you record neurons in sensory cortices, that they basically receive excitatory and inhibitory inputs, and that those often share a very similar stimulus tuning. Okay? So they're kind of co-tuned in stimulus space. If you do all sorts of tricks and record excitatory and inhibitory inputs at the same time, you kind of see by just eyeball metrics that these two things here are very highly correlated. This sort of a picture of excitatory and inhibitory inputs to that cell, so they're correlated in time. If you record from different cells, then those cells that receive a lot of inhibition or receive a lot of excitation often also receive a lot of inhibition. So there's a high correlation from neuron to neuron. And other people have, basically imprinted arbitrary patterns or stimulated arbitrary patterns in the brain, and that sort of suggests that there's also a strong correlation between excitation and inhibition across sort of activation patterns that you can generate. And so, in a way, we kind of know it's there in many ways, and so we can just go ahead and take circuits and optimize them just for EI balance, not knowing why this should be functionally relevant at all. And so we've been a bit on a lucky streak with that, if you wish. So we've been able to show that you can just optimize for EI balance and get things like place cells and grid cells a couple of years back. You can optimize for EI balance and explain sort of synaptic fine scale circuit structure in mouse visual cortex. You can use that balancing for excitation and inhibition to generate prediction error neurons, and that's something that other people have also worked on, like Juliana Georgieva has just had a paper, I think, in eLife that again went in that direction of using inhibitory plasticity to generate prediction errors or to learn prediction error coding. And so finally, and that's the story that I'm going to talk about here, is something really more about sort of generating interneuron classes in cortical circuits, basically by balancing excitation and inhibition. Okay, so and the starting point here is as I said, this idea that excitation and inhibition could be correlated in space, in time, in all sorts of dimensions. And the dimension that I will focus on here is sort of the dimension of the extent of the neuron. And so, as you know, these neurons are quite complicated structures morphologically. And so the assumption that I will make is that basically excitation and inhibition are not only balanced on average, but that within sort of different subcompartments, like maybe the perisomatic domain or the distal dendrites, even on that level, there is a balance of excitation and inhibition. Okay, and so that's basically the thing that I will try to optimize, and I'll try to show you where that takes us. So maybe a little bit on what exactly we did, or what Joram did more precisely, right? So we have a model of pyramidal cells, right? And we take not only one of them, but we take a population of those. And those neurons are now, start simple, have two different compartments, which is basically a somatic compartment that can generate spikes. And it has a dendritic compartment, or these cells have a dendritic compartment up here that can basically also show sort of active processing and generate dendritic plateau potentials. And they in turn generate bursts in the soma, and there's all sorts of fancy stuff happening. And I'll show you a little bit more about what exactly is going on there a little bit later. Now, these cells, or this population of cells, receives two inputs. One is bottom-up input or sensory input that targets the perisomatic domain, and top-down input that goes to the dendrites. And importantly, of course, for this workshop, this is a spiking neuron model. Right? And so what we did then is basically that we just took a bunch of interneurons. They're all the same, as you'll see in a second. We let them receive input from the pyramidal cells, and then they're supposed to provide feedback inhibition to these pyramidal cells. And this feedback namely goes to both compartments. And what we try to do is to balance excitation and inhibition in both compartments separately. And then we just optimize. Okay, and so we do a gradient-based optimization of excitation and inhibition both in the dendrite and in the soma by, basically optimizing the parameters or the properties of this inhibitory population here, trying to see how far we can get towards biology. And so because this is a technical audience, I'm free to just show the objective function. So the idea is really that you have excitatory inputs to the soma, you have inhibitory inputs to the soma. Those are negative, those are positive. So you just basically take the difference between excitation and inhibition, take the mean squared error over time and over neurons, and optimize for that. And the same thing is just happening on the dendrites. Okay, good. Now, a few things that sets us apart from probably most neuromorphic stuff. The pyramidal cells are now, a complicated structure with 2 compartments and all sorts of nonlinear dynamics. So this is not the vanilla flavor integrate and fire neuron. The interneurons are vanilla flavor integrate and fire neurons. The other thing that's also a little bit more fancy, if you wish, or more biology, is the fact that these synapses here that target the interneurons now undergo short-term plasticity. Okay, so they're a little bit more complicated than a standard sort of current-based or conductance-based synapse. So those of you who are not so familiar with short-term plasticity, it's basically, I alluded to that earlier, if you basically trigger spikes in some synapse and record the postsynaptic response, then what you can see is that basically in some types of synapses, you have a very strong first response, and then the second, third, etc., sort of ease off. So the synapse gets less effective with additional spikes. If you let it sit for a while, nice and quietly, then it can recover to where it was before. So that's a depressing synapse. This here would be a facilitating synapse, where you'd have the first response being comparatively small, and then it's sort of rapidly increasing and plateauing at some higher level, or maybe even declining again. But initially, you basically get a synapse that is getting increasingly effective with time. Now, there's a standard model for that type of thing that was invented by Michel Zodix and Henry Markham sometime in the late 90s. I won't go into the details of what that model does. I will only sort of highlight the parameter here. This is this parameter here, U, which is describes something that one could call the initial release probability, and that tells you if this is going to be a facilitating or a depressing synapse, right? So if you make the release probability very high, close to 1, you get a depressing synapse. If you make it close to 0, you get a facilitating synapse. And so what Johan then did is he basically optimized the circuit, that is the weights of onto the pyramidal cells and the initial release probability of the incoming synapse, and I think also the incoming synapses, basically to minimize the mean squared error between E and I, excitation and inhibition in the soma and in the dendrite. Basically, because this is a recurrent circuit, you'll have to backprop through time, right? Nicely, this is, of course, as all of you know, by now very simple. You just program everything in PyTorch and use automatic differentiation. And because it's spiking, you also need to basically think about surrogate gradients. And at that point, we were quite grateful that Friedemann and a lot of other people had worried about that problem for us, and we didn't have to, because we probably couldn't have solved it as nicely as you did, right? So that's the setting. Everything works. So what you see here is basically CE excitation inhibition correlation nicely creeping up over the course of optimization. It works both in the soma and in the dendrite. So for once, a workshop where I can show learning curves, which is nice. But the more interesting thing is, of course, to see what is the outcome. And so let's start with what we have before learning. As I said, it's a inhibitory population that's totally homogeneous, right? And so the way I'm plotting this here is that I check for every neuron in that population how strong its synapses onto the dendrites of the pyramidal cell, onto the dendrites are for the pyramidal cells, and how strong its outgoing synapses are onto the soma of the pyramidal cells. Okay? And we initialize this circuit sort of randomly. So every cell sort of lies in some random bulk here. There's no specialization or so apparent at all. And the other thing that you might see here is that all these points are gray. Right? That means that the sort of paired pulse ratio, which is a measure of whether the synapse is facilitating or depressing, is 1. That means these synapses are basically boring in time. Right? They don't change very much over time. And that's also depicted here in this histogram. So basically, take home here, all the interneurons are the same. And then you go ahead and you optimize. And there are a few striking things that pop up immediately. One thing is that you'll see here is that these points here, which were previously just a Gaussian blob, if you wish, are now hugging the axes. Okay? That's one thing. So you basically have some neurons that very preferentially target the soma, and others that very preferentially target the dendrites. And they also have obtained color, right? So that means the ones that target the dendrite now have a paired pulse ratio that's larger than 1, so they're facilitating, and the ones that target the soma are now depressing. And so, and that's something that's very much akin to what we've seen in cortex. If you look at the distribution of these paired pulse ratios, you see that this is now very much a bimodal distribution. This is not sort of a continuum of things, but there are really 2 classes of cells, one of which is depressing and one of which is facilitating. And if you think back of what I sort of told you in the beginning about these interneuron circuits, this looks very much like these two cell classes, parvalbumin and SST cells or somatostatin cells that basically target the soma and the dendrite respectively and have depressing and facilitating synapses. And this is something that I thought was actually quite exciting because it's one thing to take an interneuron circuit with many different cell types and then optimize the parameters in the system. But it's yet another thing to go ahead and basically start with something that does not have that level of differentiation and then basically generate that differentiation into cell classes from scratch. Right? And that's something I found quite exciting. And of course, that sort of, let's say, would be the ultimate dream that we look at these cortical complicated circuits and that we don't have to model them by putting in that there are cell classes or what they look like, but just say like, hey, here's the computation and here's the circuit that comes out and it looks exactly like cortex, right? That would be sort of a long-term vision. Whether that's realistic is I guess, a different question. Okay, so something that might be interesting to some of you as well, because is the question of what the activity patterns in these circuits actually look like. Because often when you do optimization in spiking circuits, the outcome is not necessarily very brain-like, right? So at least in our hands, when we try to train spiking networks for decision making, they found weird hacks, right? That they really didn't look like cortex. In this case, it's a little bit more sort of friendly, if you wish. So if you look at the currents here, you see that now excitation and inhibition are correlated both on the soma and the dendrite. But more importantly, if you look at the spikes, this sort of looks like an asynchronous irregular state for the activity of these circuits that we have now optimized. Now, I have to be a little bit careful here because the circuit is very small. If you want to backprop through time through a recurrent spiking network, and you don't want to do that on a supercomputer, then you want to keep the circuit comparatively small. And so that means we can't really talk about an AI state here because that just requires more neurons. But I do know from larger networks that we have run, and that's now work from Philippe Kreusse, who's a postdoc in the lab who basically just worked on plasticity. We know that if we basically just balance excitation and inhibition through a slightly different objective in dendrite and soma separately, putting these two cell classes in, that we actually get an asynchronous irregular state. And that's basically shown here, but I guess you see the raster plots, right? It kind of is an asynchronous irregular state, and not only for spikes, but actually also for bursting activity in this circuit, something that you'll see is quite important in a second. And so the CV of the interspike intervals is somewhere around 1. And that's something that we've seen quite often, that if you go for EI balance, that this is sort of just a network state that emerges quite robustly. And we've known that for probably a good 10 years from work where Friedemann also contributed together with Tim Vogels. Good. So, of course, as a biologist, an important question is to try to understand why this looks like that. And so we try to understand what the underlying mechanism is. In a way, now I'm telling the story a little bit in a reverse chronological order, because the reason why this emergence is actually something that we had guessed before, and that is now work from Richard Naud. And Richard, I saw you're here. Great to sort of check up on what's going on. So the challenge that the system is facing is the following. So you have a bunch of pyramidal cells. They receive input to the soma. They receive input to the dendrite. And then they just sort of crunch all of that together, right? And generate output spikes, right? And I made them green now because, of course, the output of those neurons, depends on both this yellow stuff and this blue stuff. And what comes out is some kind of mixture, right? Now, the challenge, if you now want to balance excitation and inhibition in the soma and in the dendrite, is that you basically need to unmix that mixture. Right? Because if you want to balance excitation and inhibition in the soma, you need an amount of inhibition that is in proportion to the amount of excitation that you've received from the outside. And the same thing is true for the dendrite. You need an amount of inhibition that is proportional to the amount of excitation that you've received from the outside onto the dendrites. But now, if you want to do that through feedback, right, basically feedback inhibition through projecting to the interneurons and coming back, then the interneurons somehow have to find out what the blue stuff and yellow stuff was, only knowing the green stuff. Okay, and that's is an inherent problem, and a problem that we had looked at before, basically coming in that direction from a different perspective. And that's something that Richard termed multiplexing, where these pyramidal cells actually encode both the yellow and the blue stuff by sort of exploiting spike timing. And I'll show you how that works. But to get there, we'll have to dig a little bit into intracellular processing in these sort of more extended pyramidal cells. So something that is known for probably a good 25 years or a bit more than 20 years now is how this looks if you look at, let's say, layer 5 pyramidal cells. So there's bottom-up input that targets the soma, there's top-down input that targets the dendrite, and there's some kind of output. And so what Matthew Larkham has shown in the late 90s is basically the following phenomenon, that if you target the dendrite alone, Matthew is the master of dendritic patching, right? So if you just go ahead and stimulate the distal dendrite, then you get these sorts of events in the dendrite, much weaker in the soma. And then if you stimulate the soma alone, you can trigger spikes, which is nice. And in the dendrite, you see these sort of elevations in the voltage because there's backpropagation of the spikes into the distal dendrite, which has sort of this little sort of plateauish thing at the end, but it's still one spike. But if you combine these things, if you give an input both in the soma and in the dendrite, then suddenly something magic happens, and the dendrite basically crosses some kind of threshold and generates this long sort of plateau potential in the dendrite. Okay, and what this plateau potential generates is, because it also drives the soma, is then a sequence of spikes that emerges from some kind of ping pong between dendrite and soma. And so the picture that emerges from that is that there is a nonlinear interaction between somatic and dendritic input that can generate these types of bursts. Now let's abstract that away a little bit into a cortical code or a neural code. Right? So basically, let's assume that there is a population of these types of cells. Right? And so the assumption I'm making is that somatic input generates spikes. That's basically this part of the picture here. That dendritic input converts these spikes into bursts, and I will assume that there's some kind of background noise, so it will do that stochastically. Right? So if I can make that assumption, then I can basically think about how the population will encode somatic and dendritic input. Somatic input will generate events. Those could be either single spikes or bursts, right? That for some reason have been turned, or spikes that have been turned into bursts by dendritic input, or and those I'll call events. And then the dendritic input is basically the proportion of things that happen that are turned into bursts. Okay, so that's basically this assumption that you can turn spikes into bursts stochastically. And so if that's the case, then in the end, if I look at the spike trains of that population, I could try to find out what the somatic input and what the dendritic input was, just looking at sort of the number of events and the burst probability or proportion of events that are actually bursts. And so both of these things, both the bottom-up input and the top-down input, are then synchronously encoded by that cell or by that population of cells. And that's something that Richard Naud then instantiated nicely in simulation and has wonderfully exploited over the last couple of years. And so the idea is really that it can be depicted here. So if as a function of time, somatic input increases and dendritic input maybe decreases, then in the beginning, it's maybe some first time window, you have a lot of dendritic input, so the spikes will all be converted to bursts, so you might be getting a burst here. In the second time window, where it's kind of half-half, you might get one spike, a single spike, and one burst potentially. So you'd have two events, but one of them would be a burst. And here in the last time window, dendritic input is low, so it's mostly spikes. So you'd have two single spikes and one burst. So that's three events, one of which is a burst. Okay. So if I put this into the code, basically I'm ending up with just one burst out of one event, which is a burst probability of 100%, 1 out of 2, 1 out of 3. So I get a decline in the proportion of events that are bursts. And that's then sort of a mirror image of this dendritic input here. And so on the other hand, if I don't care about the additional spikes and the bursts and I only count the events, then I see that there's 1, 2, 3 events, and I get an increase in the amount of spiking, and that then mirrors the amount of somatic input that's coming in over here. Okay, so that's sort of the idea. And if you simulate that, then you see that this idea actually works quite well. So this is a stimulus to soma and dendrite that's designed such that you basically don't have much signal so that these two inputs actually vary over time, but that the overall firing rate of the cell or of the population doesn't vary very much. But if you now resolve things into bursts and events, then you might see that sort of the event rate here is a very nice mirror image of the somatic input up here. And the burst fraction, so the proportion of events that are bursts, is a very nice mirror image of this red line up here, which is the dendritic input. So that means you basically encode both of these things at the same time if you look at the temporal patterns that these neurons actually generate. So that's basically saying that just the biophysics of pyramidal cells basically allow you to encode bottom-up and top-down signals simultaneously by sort of, exploiting spike patterns. That's the idea here. And so, of course, the question is, that's just encoding. So let's see how decoding works, right? And I'll promise you to come back to the interneurons later on. So the thing that I think is easy to imagine, if you have such a population of cells and you would now be interested in the somatic input, then that should be basically just the number of events, meaning what you have to do is you have to count the number of spikes that these neurons emit and discard the additional spikes within bursts. Okay? And the simplest way to do that is just to use a depressing synapse. Right? So you just basically, a depressing synapse would, within a burst, count the first spike and then sort of get less and less transmission for the additional spikes in a burst. So the postsynaptic current would be a reflection of the event rate rather than the firing rate of that population of cells. Right, and that's something that Richard could show. So if you increase the amount of somatic input, then the population rate of a downstream population of neurons that receives depressing synapses is basically nicely increases with somatic input. And these different lines here are different amounts of dendritic input. So you see some kind of invariance to dendritic input, which means you're kind of unmixing things. And so the other thing, of course, that you need to decode is the dendritic input. And as I said, dendritic input generates bursts. So, and I guess you can now already guess what's going to happen. So you basically need facilitating synapses, because if you want to measure bursts, then you're not interested in the first spike, but you're really only interested in the second, right? And the second in very short succession is an indication that there was a burst in that specific cell. And so you'll have to be a little bit more careful. So you need a facilitating synapse onto this, or synapses onto this population of neurons. There's a little thing that I won't talk about very much, is that you at the same time need some kind of feed-forward inhibition, and maybe you just postpone the discussion why this is something that is useful. Because it's actually something that we thought for a while was a divisive normalization. I'm no longer so sure about that. Okay, so that's just the take-home here, is that you can use short-term plasticity to decode these two streams of input that impinge onto the pyramidal cell population. Now, if I come back to my interneurons, then the optimized circuit ends up doing exactly that. Okay, so if I have my population of cells and I have one interneuron class that inhibits the soma, then it should be active, or its activity should reflect the amount of bottom-up input to this population of neurons in order to achieve a balance of excitation and inhibition. And that means it should receive depressing synapses. Okay? And similarly, if this population here targets the dendrites, and it should basically provide an amount of inhibition that is in proportion to this top-down excitation that arrives here, then it should be representing just the dendritic input. So it basically faces the task of decoding from the activity of the pyramidal cells the amount of dendritic input. And that is something that you can then do through short-term facilitation. Okay? And this is basically the circuit that we get out through the optimization procedure using surrogate gradients. Okay, little side note, this feed-forward inhibition we also see, which doesn't map so nicely to cortex, but that's probably more for the aficionados. Okay, I think it's time to summarize, if I'm not mistaken. So what I try to bring across is that, optimization in spiking networks is sort of a nice way of just restricting parameters, not only in terms of engineering problems, which were very much the focus of the last session, but also in terms of just restricting parameters in models of biological circuits, right? And so my personal hope is that at some point we can get to a situation where you can just optimize circuits, and then they look in the end like the brain. And then that tells us something about what the computation is that the circuit might be doing, because of course we have to define the objective in the beginning. At the moment, the objective that we chose was excitation inhibition balance. As I said, I don't believe that is a goal per se. I believe that there is a lot of computations that profit from excitation inhibition balance, but I think that's a longer debate where I'm not sure if I have converged on my own opinion. Second thing is that's more for the biology people here, that you can basically generate the diversification into interneuron classes from basically the need to decode a multiplex neural code for somatic and dendritic inputs. And maybe something that I didn't touch upon very much, because that's Richard Note's game, is what this multiplexing is actually good for, right? One thing is that you could potentially flexibly combine different signals, which I think is an interesting path to pursue. But something that Richard has very, very nicely illustrated is that this multiplexing might allow to actually process two information streams in parallel, and specifically a bottom-up stream of information and a top-down stream of information. And all of you are, of course, familiar with that type of challenge of backpropagation of error for learning, which is a backward sweep, right? And basically the simultaneous processing of information, which is a forward sweep. And so, of course, then having two signals that travel through a network in parallel is quite an exciting opportunity. And together with Alexandre Pailleur, Richard, and quite a few other people have recently illustrated that. Okay, with that, I'm going to close. If you want to read more about this, then there are basically three papers out there for you to read. One is the multiplexing story, which is now 3 years old. That is in PNAS. The idea of diversification into interneurons is more recent. It's still a bioarchive paper. We hope that it will eventually be out. And sort of balancing and this sort of doubly asynchronous irregular state is recent work from Philippe Kreuze that is to appear in PLOS computational biology. And with that, I would like to thank you, of course, first and foremost, for your attention. Of course, all my funding sources, and specifically, of course, Joram and Richard, who did all that work. And yeah, thanks a lot for inviting me. And I wish you all, I'm looking forward to your questions.
Great. Thank you so much, Henning. So yeah, we're a little bit short on time, but there's many questions. And actually, the first question is maybe Tim, who can ask himself actually his question. He's on stage right now.
Okay.
Hi.
Can you
Yes,
hear
I
me?
can.
All right. It's actually the second question with more votes. The first one was by Dan, so I shouldn't be here. But okay, it's a very simple question. But yeah, the excitatory inhibitory balance is your objective, but I'm surprised it is constraining enough. Can't you find many solutions?
There are many solutions, right? But in the end, the problem is, let's say, comparatively low dimensional, right? Because the issue is that, of course, all the excitatory cells and the all the somata of the pyramidal cells basically receive the same input. We think of an ensemble of neurons that all code the same thing, right? And the same thing for the dendrites. So in the end, it's really just a two-dimensional stimulus space. So you can think of these sorts of as homogeneous populations. And of course, every run will do something different in the details, right? But we're basically in a mean field game, right? So the computation is, if you wish, a halfway trivial one, right? Making that, of course, a high-dimensional input and high-dimensional output, both on the soma and the dendrites. Then, of course, the thing that you're mentioning, there could be all sorts of stuff happening, and I don't dare to speculate.
All right.
Does that answer your question?
Yes, you did. Thanks.
Okay. Thank you.
Thanks a lot. So yeah, the question that got most votes is actually Dan, but I think he cheated by posting it really early into the talk. So I'm just quickly going to read it. So would you agree with the Richards et al. Deep learning framework for neuroscience paper approach? That is, that the right level of design is to look at the objective functions, learning rules, and architectures.
And objective functions, learning rules, and architectures. Yeah, but isn't that almost everything? in the end, sounds stupid, right? But in the end, what do you mean by architecture, right? I mean, the objective function is what the computation is, which is, of course, super interesting. That's what we're all interested in. The learning rule is basically a way of getting there. And then the architecture is how much you put in from scratch, from biology, right? And so that's, if you wish, some kind of inductive bias or so that you would put in. And so the question is, where do you stop there, right? Every constraint that comes from biology, to me, can be sold as architecture. So in that sense, I can agree with that, but I'm not sure if that's a very strong statement. Is that an answer to your question, Dan?
And Dan actually has to prepare the next session, so he
won't be here. Okay.
Later, but I think you could have just answered with yes. It seems like you agree, yeah. But yeah, I think it captures a lot. But I think and I think that should answer his question. So it's great. So there was another question by Marcus Gosch. Thanks for the great talk. One thing I wasn't sure about was why you preferred or referred to EI balance as a straw man.
I refer to it as a straw man because I wouldn't immediately know why there should be evolutionary pressure for excitation inhibition balance. Right? So in the end, I think the whole circuit is supposed to do some kind of computation. Right? It's supposed to make decisions, control muscles, perform sensory inferences, or something like that. That is something where I can immediately see why there should be some kind of evolutionary pressure to do these things well. Right? And so in the end, I think that might entail in the end the circuitry that exploits excitation and inhibition balance for something, for some kind of purpose. But it's something that is, for me, a little bit on the way. And I wouldn't even know if it's something that is true everywhere. And we know it's not true everywhere, right? There are systems where EI balance is just not true. So in that sense, I think we're using something that we see a lot in cortical circuits. It is surprisingly effective to get out stuff that looks like the rest of cortex. And I personally don't quite know why. But it's something that I wouldn't, use as an objective per se if I wanted to design a system, right? I mean, if somebody from the engineering perspective comes in and designs a network, then excitation inhibition balance is no objective that makes any sense. Do you know what I mean?
Yes, I hope Marcus also knows what you mean now. Thank you very much. So there was another question which triggered a large discussion actually by Chris Curran. And so what he writes is there's recent experimental evidence that by the time inhibitory neurons have migrated and connected, they already have the genetic expressions of the cell class. Do you think this is a result of evolution solving the EI balance so that the brain doesn't need to worry about it in its lifetime?
I would love to pass that question on to Yoram if I could, because he's actually dug into that very much. Because it's basically a question that then sort of relates to nature versus nurture, which is course.
Maybe I should point out that what Yoram posted extensively.
Yeah, I can imagine. Yeah, I mean, Yoram has gone very deep into exactly that type of question. But you can ask these questions by or try to address these types of questions by saying, how far back can you go in evolution until sort of a dendrite appeared, and how far back can you go in evolution where the first hallmarks of of things like, for example, synaptic facilitation appeared. So you can just try to see if there's some kind of co-evolution of these things, and it's a surprising adventure if you go down that route. And I think that's something we should defer here a little bit, because Yoram, I'm not sure if I can paraphrase Yoram's insight well enough. I think he has some kind of opinion piece for that in the early stages of preparation. So I hope to be able to answer that question more thoroughly at some point.
Very cool. Thank you. Good. One more quick one, perhaps. So this is my own question. I'll skip that. Thomas Novotny asked, maybe I missed it, but what were the assumptions about the homogeneity or otherwise of the pyramidal cells in the model?
Well, they're electrophysiologically homogeneous. The inputs are homogeneous, apart from noise, right? You need to add some kind of noise to generate some kind of fluctuation. What else? What else could be homogeneous? I think that's pretty much. You can play a little bit with how you constrain certain weights in terms of how much freedom you allow. But that's something that is more a question of how quickly things converge. And correct me if I'm wrong, Yoram. or if you actually, how large your search space is, if you wish. But the restriction of homogeneous input and homogeneous electrophysiology is, of course, quite an important one, and it's clearly not true, right?
Cool. Right. So 30 more seconds. So I'll squeeze in my question now. So you optimized the U parameter basically of the short-term plasticity model, which basically gave you the spread between the facilitating and depressing. Did you also look into optimizing the time constants, and do they align with anything that you would expect from
the energy landscapes for the time constants are not very well behaved. So one thing, at least that's the way I remember it when we played with it. Joram, feel free to add in the chat. But if I remember correctly, we tried that. But don't try to optimize the time constants. If at all, optimize the rate constants, right? So the inverse time constants, that's a little bit better. But even there, and even the short-term plasticity itself is something that is in some parameter ranges very insensitive to these time constants. So it's not easy to optimize those.
Okay, thank you. Thank you so much. There was one more quick question which I'll copy and post in the chat. Unfortunately, we're out of time. Maybe you can answer it in the chat by Louis Riquelme was this question. So thank you very much, Henning, and thank you very much to the audience for the great questions. And now it's time to move on. So I'll end this session. I will see each other with Viola Friesemann in the next talk. Thanks so much. See ya.
Thank you very much.